Good morning. The exam, of course, is on Tuesday. For those who were curious about its makeup and just came in just a moment ago, it's in 4 parts of pretty much equal weight. This is multiple choice. It is diagram. It is matchup, and it is true and false. Nothing much more to add about this, except for those who are taking it into pairs. I will drop the copies off in the morning before I come here. Moving then onwards and finishing up this chapter on seeds and seedling biology, I mentioned that seeds are typically go into a state of dormancy. Not all, mind you, but we should be looking at this from the standpoint of a life cycle now. When we get to the seed plants, it's not enough to go into this is what these are gymnosperms, these are angiosperms, etc. We have to look at these organisms as sharing a life cycle. So we're about to move into germination. We will then today pass on to primary growth and what this contains. And then in weeks to come, we will be looking at the transition from primary growth to secondary growth. Secondary growth leads to sexual maturity, whereas dispersal is the end product of sexual maturity, and there may or may not be this period of dormancy. Some plants do, some plants do not. So we understand then that breaking dormancy will differ in different species of plants. In some, it's just a matter of adding water, as you learned in lab yesterday, some of you, and then the whole process begins. Others, there are barriers in between based on what kind of environment they are adapted to. When we mentioned slow erosion and mechanical cracking after cyclical freezing, animal action, there's probably a lot more of that than people have actually worked on. Is the seed coat worn down by stomach acids or mechanical movements in a crop or gizzard? And then we've also seen this situation in cyclical fires. That's because certain seeds contain anti-germinants in their seed coats, and the seeds do not germinate until these anti-germinants in the form of tannins are removed. Sometimes this requires a long period of soaking or some sort of mechanical action. So in the act of germination, we should expect 8 stages in the act of the seed escaping from its coat. However, when we take a closer look at monocots and eudicots, there may be as many as 2 extra phases that may go in here. But the first thing that we expect to see is that the first act of germination is, of course, that the tissues inside have to rehydrate. Water in some form has to reach the embryo. With the absorption of water, the tissues expand inside, the cells get bigger, so then obviously the next phase is that the coat will rupture, the seed coat will rupture. It can either occur along straight lines of sutures, or it may fragment, as some of you have already seen in lab. Once the seed coat ruptures, we go into stage 3. Oxygen now reaches the tissues, and the embryo switches from anaerobic to aerobic respiration. Under 4, the radicle emerges. Let's make a point that in the majority of seed plants, what we expect to see occur during the early phase of germination is that the first part of the embryo that leaves is the radicle, which is basically the tip of the hypocotyl. This seems to occur in the majority of cases. What should happen next, then, is that the epicotyl emerges and undergoes a period of elongation in some way, shape, or form. It's going to stretch. By stage 6, the cotyledons will usually expand and provide initial photosynthesis, particularly in the eudicots, as you've noticed. We don't expect that much in the monocots, which means that by stage 7, the cotyledons have pretty much done what they're supposed to do. They're going to wither and drop off, and we expect that by this point, the epicotyl is allowing the young foliage leaves, the plumule, to expand. What we should therefore see by stage 8 is that any remainder of endosperm or endosperm nutrient is going to be metabolized by the plantlet. Sorry, what? Sure. By stage 8, any remaining endosperm that may have remained inside the seed coat is gone. It has been metabolized. It has been used up, and there are some unique ways in which this may occur. It's here that we get a little variation. In monocotyledons, the cotyledon absorbs metabolized endosperm and then passes it on to the plantlet. As I said, in monocots, the cotyledon is not usually photosynthetic. It's serving as a modified stomach, and it's not contributing to early photosynthesis. All it's really doing is channeling the sugars that are coming out of the endosperm, which is breaking down. When we look at germination, then, we expect to see two different forms of germination dependent on species. And what most people are familiar with, if you grow your little radish sprouts and this sort of thing, is that we expect that the seed, as it germinates, produces a hypocotyl, which expands and pushes the seed above the soil line. At some point, we expect the cotyledons to push the remainder of the seed coat off of the epicotyl. So what we have here is a state in which the cotyledons actually reach the soil surface and expand. This is epygean germination. Epygean, on top of Gaia, the earth. The cotyledons are above the surface of the soil line. The exact opposite, which we also have in lab, is that the seed once again produces the hypocotyl. The hypocotyl, however, doesn't expand and push things up. It stays where it is, which means that at some point we have to expect that the epicotyl emerges on its own below the soil line, and then at some point pushes out its little plumules. It means that the cotyledon or cotyledones, it can be either in a monocot or a dicot, the cotyledons remain underground and never reach the soil surface. This then is hypogean germination, and you can have both forms of germination in rather closely related plants. Beans tend to be Epygean. Peas, on the other hand, for those who haven't looked at it yet, tend to be Hypogean. There is, however, an extra stage in certain monocotyledons, particularly in grasses. In these systems, specifically in grasses, we normally think of a typical Hypogean system like so, with the cotyledon still inside, acting as a stomach inside of the seed coat itself. But something new has been added. We're dealing with plants that have very thin shoots and roots, and there's this initial problem of dealing with the sharper grit within the soil. These plants tend to have a very specialized sheath surrounding them, which protects them as they move upwards or down. On the upside, we speak of a coleoptile protecting the shoot and a coleorhiza protecting the root. But this can't go on for too long because the sheath is preventing the structures from expanding and dividing. The coleorhiza, if it were to stay, would mean that you could only make one root. So what we expect to see in these systems as they get older is that we expect that the coleoptile peels off at an early stage and resembles a little turtleneck sweater on the young stem, and the coleorhiza does pretty much the same on the underside. It pulls back as a flap, allowing the stem to produce its plumule and allowing the root to undergo secondary growth and produce new roots from inside it and to branch out and to become more fibrous. We'll have more to say on root development in a later lecture. So what we see within the seed plants is a tremendous difference in the physical size of the seed, which I've in fact placed in one of the handouts you got on Tuesday. Seed side, then, is going to be largely determined on the amount of endosperm, or at least food stored inside, certain cases, starchy cotyledons. As I said, in the legumes, as in the beans and peas, the endosperm is digested very early, and the food winds up inside the cotyledons, which is why they're so plump and look like little catcher's mitts, fleshy catcher's mitts, when they open. In other plants, though, we expect that endosperm makes up the majority of the interior of the seed. The embryo is quite dwarfed by the amount of endosperm inside, but there are exceptions to the rule. Who produces the biggest and largest seeds? Gymnosperms, monocots, or eudicots? Somebody. Sorry, what? No, and in fact, it's on your handout on the last page, got you all, that it is in fact the monocotyledons that produce both the biggest and smallest seeds in nature, the largest of which is pictured on that young boy's shoulder. It is in fact in one of the palms. It is the coco de mer. This is a species only found in the Seychelles Islands of the Pacific Ocean. It can take up to 6 years to mature on the tree. The trees are in no rush before they fall off. You can see that what you're looking at is actually the fruit wall. The seed is about the size of a regulation football. So large that the seed coats were used to make ceremonial drinking cups. But palms in general, what we think of, just say a common coconut, actually bears two kinds of endosperm. It's so big. There is the solid cellular endosperm, which we grind up and use as the coconut meat and various things. But do you realize that since these trees are normally dispersing from one beach to another naturally by floating, that they're also taking, since they're on sand or grit, water drains very quickly. So what you think of as the coconut water inside of a coconut is the enzyme-rich water they take with them in order to germinate. This means that coconuts can, at least in their early phase, colonize areas in which rain may be infrequent, or in fact rain is quickly lost because you're on a sandy soil and it drains away. The smallest seeds in nature belong to the family Orchidaceae, and it seems to be pretty successful because there's over 20,000 species of them, and they virtually all do this. If you look at the drawing or otherwise, it's this simple. You have a seed coat that's just a couple of microns long, and it is only one cell thick. Looks like a little bunch of bubbles all attached to each other, whereas inside there is a smaller embryo with the typical monocot notch as usual, but there's really nothing much in the way of a hypocotyl. What has to happen with an orchid seed, and they're pretty successful, these seeds can float in the air and remain viable for about 1,000 kilometers once a wind blows them away from the parent plant. But push comes to shove after they land, because they cannot survive unless they meet up with the proper endomycorrhizal fungus and the glomeromycota, you will remember. It's at this point that the embryo escapes from the coat here, but it's such a tiny structure you can actually count the initial number of cells within the embryo, because they're usually only about 1 cell thick. People talk about those that have 34 and those that have 42 and this sort of thing. But they then generally grow into these little top-shaped structures referred to as protocorms. And instead of making roots on their southern end, they just produce a bunch of root hairs. They're photosynthetic at this point, but they're waiting for a fungus to attach itself to a root hair and then, in fact, invade the cells and produce arbusculae. This is often a rather slow process, and a protocorm may not produce its first true foliage leaves. Its plumule don't do much. It may not produce its first plumule leaves or its first roots for anywhere from 1 to 5 years after they meet up with the fungus. Which takes us to the other matter of human nutrition of seeds, which in fact is also a monocot affair, because most of the seeds that are consumed as a source of carbohydrates are grasses, and grasses are monocots, right? Okay. A wheat kernel, then, is not what you think. It's actually a specialized fruit with a thin fruit wall covering a single seed. If we were to look inside or corn will work just as well, but we'll make this. What we actually see here is a double layer. It's a single-celled fruit, but what's happened here is that the ovary has fused to the seed coat and would be referred to as a pericarp because it's a combined structure. And inside there is a teeny-weeny little embryo with a larger cotyledon or scutellum, and this is then surrounded by a lot of endosperm. But in between the endosperm and the pericarp, we expect to find, some of you saw it in lab the other day, there is a specialized chain of cells called the alurone layer, which we tend to see in grasses, but we don't necessarily see it in other plants, not even in other monocots. So when we think about what we're getting out of a wheat seed, endosperm is what becomes flour. In its original state, until it's bleached or something, then the endosperm of a wheat kernel is anywhere between 9 to 14% protein, 86 to 91% starch. And that takes up the majority of the mass of any wheat grain. Wheat grain is its mass consists of about 80% endosperm. What do we mean when we go into Whole Foods and they rip us off because we're going to buy an overly expensive jar of wheat germ? Wheat germ, then, is the embryo. It has been removed from the seed. It has been detached from the endosperm and has usually been toasted. The toasting process is done because it's oily and full of vitamins, and if you let it sit in the raw state, it goes rancid very quickly, but this only consists of about 5 to 6% of the wheat germ. Now, how many people started their day with a bowl of cereal that contained bran? Ooh, a lot of liars in here. Okay. Bran, then, is the indigestible pericarp. It is the alurone, it does contain the alurone layer, which should be digestible, but since the pericarp has been separated from the endosperm and the embryo, and since it's crumpled up, whatever is available in the alurone layer is not available to you in the form of bran cereal. Your small intestines can't get at it because it has an indigestible seed coat wrapped around it. This makes up about 14% of a grain of wheat. The alurone layer, however, is extremely important to the embryo itself, because in these grasses, the alurone layer is embryo-rich when it is time for the seed to germinate, when it is time for the hypocotyl to emerge. It becomes biochemically very, very active, produces these enzymes which start to break down the endosperm, making it much easier for the scutellum to digest. We don't get the benefit of it. The diet fads for bran that went on in the 80s and 90s were simply telling you, whatever you eat, throw a lot of bran onto it, because this slows the digestive process, and your small intestines may not absorb all the fats or sugars that are going through at the time. That didn't last, obviously. Now, once we have these organs emerging from the seed, the seed plant has now switched into another stage in its life cycle. It is going to go through a period of primary growth. This period of primary growth can also be referred to, at least initially, as establishment. That is at least the early phase of primary growth, and it means what it says. When we talk about establishment, we are talking about a period in which the radicle establishes its early root system in whatever it's penetrated, whatever it's growing in. It means that the epicondyle establishes its early stems and leaves. It marks a period of active growth and development that will later culminate in the mature vegetative form of the species. During the process of establishment, immature growth can be recognized in plants as well as in animals. It's not as easy as discriminating between a caterpillar and a butterfly or a tadpole and a frog, but we can, with certain plants, see at least 5 characters that we look at and you say, oh, it's showing primary growth at this point. The first is the presence of juvenile foliage. This is a big issue to farmers when to use a herbicide, because they have to recognize the juvenile foliage of certain leaves. We understand that certain plants produce a leaf form during establishment that they do not produce when they go into secondary growth. In eucalyptus, for example, we expect that the first leaves along a stem or a branch in a young tree will usually be opposite each other. They're round, there is no petiole stalk, they're a different color because they have a different wax. As the tree matures, you get a secondary foliage in which the leaves are now alternate, have a long petiole, and the blades have a sickle shape, and they're producing different waxes. Some plants have a very long period of primary or immaturity. The common ivy that's walking up the walls all over the place. That is mostly juvenile foliage with the typical lobed ivy leaf. It can take an ivy up to 20 or 24 years in this climate before it produces its mature leaves and gets ready to flower with simple, once again, usually hook-shaped leaves, and then it makes its flowers. But you can wait an awfully long time. So we understand that in primary growth, you may see juvenile foliage, not necessarily, but you can. Secondly, in these immature primary growth systems, there is a comparative absence of storage organs. Storage organs are usually made by a special group of meristems that are not active during the period of primary growth. Hence, if you plant carrots, you have to wait 2 years to get a decent carrot. You pull them up now after a single season, you get something about the size of your fingernail. So it's only in the second year that these other meristems are activated. Third, there may be differences between primary and secondary growth in the protective coverings. For most trees that are sprouting one year, they're really not replacing their epidermis with bark until the end of the season, when they start switching into secondary growth. Fourth, we do not, during this period of immaturity, see much in the way of woody growth. We don't see the plant building ring after ring of wood, if they make wood at all. And then, as this is a juvenile stage, last of all, we expect there's no sexual reproduction going on during this period. How are they doing this? How are they getting through this period of primary growth? That's what distinguishes them from animals, because when you talk about any of the seed plants, you have to talk about a primary growth that is based on meristems. So, seedling establishment is characterized by intense meristematic activity, especially primary meristem activity. And meristem is from a Greek word, merizine, meaning to divide. It refers to centers of undifferentiated tissue from which new functional cells arise. What does that mean? It means that we will expect that meristems are made up of these cells called initials. And initial has no cell wall, has concentrated granular cytoplasm, and so that makes the nucleus look a lot bigger than what it would in a differentiated cell. We have mitosis, and we expect to produce two daughter cells, and in most cases, one of the daughter cells simply stays in place and is just a new initial. Meristems are, you might say, self-rejuvenating in most plants, whereas the other will differentiate and will, at the very least, produce a primary cell wall. Some go even further and make a secondary wall. We'll get to it. But this is how you build structures in a plant. Not all of the cells have the capacity to go through mitosis, and usually, not in all cases, but usually, once a cell builds a primary cell wall around it, it can't undergo mitosis anymore. Or if it does, it doesn't last very long. It does not make too many new cells by mitosis. So unlike animal cells, if we think of animal cells reaching maturity, that's usually, there isn't too much mitosis going on after that. Now, is there? In animals, once they reach, say, sexual maturity. A lot of meiosis may be going on, but not much mitosis. So plants, when they reach, seed plants, when they reach their adult form and function, will often continue, sometimes for centuries, to undergo mitosis through division of their meristems. Which takes us to the concept of modern biotechnology of the mericlone. About 50 years ago, people began to perform some experiments on meristems of plants. They would dig them out as a little mass of cells. They would put them in a flask with a nutrient media, and there would be your little meristem. They might continue to feed it certain hormones, as we will see later on. But the first thing that happens in here is that usually it grows into a larger, undifferentiated mass referred to as a callus. What should finally happen is that if it's treated properly, the callus begins to produce little plant clones on its surface with their own leaves and their own little roots. At this point, they can be removed usually and potted up. Various industries now work this way. It's certainly good for threatened or endangered species that everyone wants, because virtually all of the Venus flytraps that you buy in nurseries these days have been cloned. They're all pretty much the same at this point, and this takes the pressure off of wild populations. When you think of it, of course, it is this industry which gave birth to modern biotechnology in the form of producing, of course, animal tissues that will do pretty much the same thing, or at least be biochemically active, and we can use the insulin they make or whatever. So you should be thanking the people who had to have that Venus flytrap or that particular orchid 50 years ago. It has applications. We can classify meristems according to the growth system they produce. Primary meristems make primary growth. They are responsible for an increase in cell number and length. In other words, they make the plant longer. Such meristems are confined to either the tip of shoots, as I placed here, or the tip of the primary root or radicle. Secondary growth is made by secondary meristems, which I will not really be mentioning for another couple of weeks. They are responsible for an increase in width and girth. The plant gets wider. It may become wider internally by producing new layers of tissue that spread it out like a tree trunk getting wider. But it can be even simpler than that. It can be buds on a stem growing, so you get dichotomous branching. Or roots may start to produce new roots along their sides, producing fibrous growth. Secondary meristems are not confined to growth tips. They may be hidden, very frequently they may be hidden on or inside the plant at various junctures. Looking at primary growth, and we will look at the roots meristem before we take our break. The radicle then is the primary root or taproot of the hypocotyl. The root meristem is found slightly beneath the tip of the primary root. It has to be protected usually at all times by a root cap, which it has to keep replacing. Remember, as the root pushes its way through the soil, there is erosion, and you've got to protect what's inside here. So the root meristem is usually located not right at the tip, but slightly back from it, and it's pushing out new cells. Okay. So the primary growth then in roots in most, but not all plants, is typified moving away from light and towards the center of gravity. Some roots don't do this. They're actually growing towards light and away from gravity, but you'll have a chance to see them in a few weeks, and they're not that common. That means that the oldest section of a root is always closest to the plant's stem, obviously. Root meristems are tiny. They are only about 10 to 20 microns in diameter, and they are comprised of many-sided initials. Each initial, as usual, has dense granule-like cytoplasm, a large nucleus, and no wall. But there are two possible forms of organization in root stem initials that we've found by sectioning. In the open type, as I've placed here, all root regions emerge from the same cluster of initials. This is an undifferentiated root meristem because you cannot predict in this one what each daughter cell is going to become. Is it going to come off here? Is it going to go here? Is it going to go here? You cannot predict what it is going to differentiate into. In the second form, the closed type, initials are layered. You can look inside and see that there is a layer upon a layer upon a layer. And we see that as daughter cells are produced, they have only one way to go. You can predict, according to the layer, what embryonic tissues, what embryonic cells they are going to make. However, there is always one part of a root meristem that consists of initials that do not undergo active mitosis. This is the inactive or quiescent center. It is usually found often slightly off-center inside of any root meristem. It does not undergo active division even when the rest of the initials around it are doing something, where they are. Quiescent centers rarely divide unless the active initials are damaged. That is, something has to happen to the root. Part of it gets ripped away. And remember, growing through soil is kind of messy and dangerous. We can think of the root cap being knocked off and the root meristem being damaged by coming in contact with the shard. It's at that point the quiescent center springs to life and starts to divide and repairs the damage. So it's basically a regeneration center upon damage in plants. Looking then at this root cap that it is making, we expect that most roots are going to be protected by a root cap. The cap is continually replaced by these acts of mitosis. So we understand that even in a layered or an unlayered system, some of these cells have to differentiate and become part of the constantly regenerating root cap. The cap contains cells that in fact contain what we would call slime. As the root cap breaks down, as the root cap is being pushed through, it's expected to wear away. It's expected that these cells are going to rupture. That's a good thing, because slime is basically a lubricant. It helps the root move through the soil unimpeded. Slime, by definition, refers to these polysaccharide pectic compounds that are stored in the Golgi bodies. In root caps, though, the Golgi bodies fuse with the plasma membrane, and they actually secrete their slime into their own cell walls, which get roughed up and broken up, and so they release it. What you're eventually going to get in the root, though, as this stuff begins to break down, is that as it's moving down, much of the root is going to become covered by this slime sheath, which is referred to as mucigel. And we found that mucigel does all sorts of things. It will contain antibiotics, and this will slow the invasion of pathogenic organisms. It may also contain chemical signals that encourage certain fungi to come in and establish mycorrhizae. And as we've seen, of course, since it's very watery, it's going to wind up keeping these growth areas moist, even if the environment around it isn't all that moist. Root meristem then produces three kinds of tissue besides the root cap itself. And we'll get into that after the break, and we will start this round. Yes. It's on the tape. There's two. And I've left the Tuesday handouts on the table.
So these are the one from Tuesday?
Mm-hmm, they're from Tuesday.
I was sick, so I was off that day.
That's all.
Do you need a doctor's note?
Sorry, what?
Do you need a doctor's note for the Tuesday?
No. Feeling better now, Mr. Fujita? Yeah. I'm underwhelmed. Okay.
Dr. Brickman,
Sorry, what?
you sent me an email about taking my test at the testing center.
Yeah.
Is it okay if I take it here instead?
With my blessings. But remember, I can't give you extra time.
That's fine.
I just, because there's someone here before and there's someone.
Yeah.
When we used to meet at the BEB, I could start people off in my office, but I can't do that now.
Yeah.
I can't be two places at once.
Yep.
Okay.
That's fine.
So I just need to turn one over on Tuesday.
Yep.
Yeah.
Show one of these hangouts.
If you haven't taken them, I suggest you do.
This is it.
There's two. There's two different types.
Perfect.
Two different species. Okay.
Thank you.
Rightio. Now, moving backwards to the root meristem, as I said, it's giving rise to initially three kinds of tissue besides the root cap. It's not just root cap it's making. It's going to build all of the cells that will eventually mature into primary root tissue. So we speak first about the protoderm, which matures to become epidermis. The procambium in the center produces the vascular tissue, and it is primarily xylem and phloem. It's going to make a cylinder. Or if you prefer, it's going to make a stele inside the root of xylem and phloem. A lot, though, is what we would call ground meristem. This is cortical tissue, which will later serve as storage. Cortical tissue is primarily going to be a tissue we call, once again, parenchyma. It's living at maturity, but its initial job is to save things up and then pass certain things on to more specialized tissues. So much for the root. Let's look at the shoot meristem. Here we have a different organization, even though there is also a quiescent center once again. In most shoot meristems, the initials are in fact in two separate layers because there's two kinds of division going on to make two different groups of tissues. The first of these is the tunica, and as you can see, that's the outermost layer of initials. It's usually about just one layer thick of these small compacted outer cells. Sometimes you get another one. Sometimes you can have as many as two. But underneath are the larger, more elongated cells that make up the corpus. So the tunica, we expect, tunica should, in its divisions, give rise to protoderm, which of course will become the plant's epidermis, but it's going to do something else. Tunica will, at least initially, set up these little bumps on the side. These are leaf primordia. They will eventually mature into leaves as they get bigger, and they're going to tie in with certain tissues made inside the corpus. The corpus, then, we expect, is going to produce most of the rest of the tissues in the plant. We are going to expect that it's going to be making a ground meristem, and it's going to be making procambium once again. But I did say that there was a quiescent center. It's usually found somewhere underneath the corpus, and it never contributes to vegetative growth even after damage. That is, you can whack away at the tunica corpus, do terrible things to it, and the quiescent center usually will not spring to life and start doing any sort of physical repairs. It's a lot like Washington, D.C. today, if you know. Now, what we should expect here, though, is that the quiescent center is usually going to form reproductive structures during a period of secondary growth. So another name for it is the waiting meristem. What we tend to see in a large number of plants is that the tunica corpus has a limited number of divisions to make. It can go through a limited number of mitoses, make a certain amount of stem and a certain amount of leaf, but there's a time for it to turn off. When it turns off and isn't doing things anymore, we expect, down here we have leaves and various buds, but we expect that the quiescent center, its first impulse is to elongate. It makes new cells and stretches them out, and then we will expect at some point, one of the last things that the quiescent center does, the waiting meristem does, is that it makes the little primordia that will eventually turn into either flowers in an angiosperm or a cone in a gymnosperm. Shoot meristems usually act absolutely opposite of root meristems. They move away from the center of gravity, but towards a source of light. Therefore, the oldest part of a shoot is that part which is nearest to the root. Now, what we are going to go into in the next lecture after Tuesday will be on the various tissues that make up of seed plants. In order to do this, we have to take a step backward, which is why I've asked you to look at Chapter 3, because this treats the cell wall, which I know you haven't had very much of, and we have to understand what's going on. Cell walls, then, are formed following differentiation of a daughter cell that's been produced by initial mitosis. We should expect that a cell wall, this primary wall, which is the first thing that gets made, is going to have different layers. So we get something like this. And a primary wall that is usually subdivided into two parts. But it's all primary wall. Primary walls, what they have in common, whether it's the inner or the outer, is that they remain relatively flexible. And if you look at your book, you'll see they are composed of tiny fibers. Think of a primary wall as being spun, as opposed to being hammered out or made. They are made out of tiny fibers, which we call microfibrils, which are tiny fibers. Each fiber is composed of glucose molecules that form a tube. So what one sees with these microfibrils are these little tubes made out of glucose molecules, and characteristically they are coiled, as you can see here. By coiling them together, and by coiling one tube with another tube, etc., we are making a material that we call cellulose, and this is the dominant organic molecule on dry land. That's all there is to it. Cellulose, by the time these little microfibrils start fusing together and coming together, makes the cell wall more rigid. Therefore, cell walls remain plastic and flexible because cell walls aren't pure cellulose. There are the cell wall is going to be inundated with various pockets of other substances such as hemicellulose and what we think of as gelatinous pectins. You cook certain plant parts for a long period of time, drain off the water, and you get this gelatin. But you can reinforce the cell wall. You can make it even stronger by either covering it on the outside with various waxes, or if there are gaps in the wall at certain points, we can in fact fill them up with tougher substances such as suberin, and I believe I've already mentioned lignin. As I said, each cell wall consists of 2 layers. The outermost has a confusing name. It's called the middle lamellae. The inner wall here is referred to as the primary wall, as it sits closest to the cytoplasm. But why middle lamellae? Well, when you think about it, when these cells are placed one on top of each other, it's this outermost middle lamellae that is ultimately squeezed into the middle in between the cells. Now, as some, not all, as some primary walls mature, they may develop a secondary wall under it. The secondary wall will also consist of a series of layers. We don't have to go into it, but we know that secondary walls are also multi-layered. It is very rigid compared to the primary wall. It is largely composed of lignin, which is pretty inflexible. Once a secondary wall forms inside of a plant cell, mitosis stops and cytoplasm usually deteriorates. The thing about plant cells with secondary walls is that they are most functional then when they're dead, which may seem like a contradiction in terms. But when you have primary walls, and even when you have a secondary wall, we find that the cell walls do not necessarily are not complete. There will be pits in the walls that make the primary wall extremely thin. We're not talking about a pore here. We are talking about a wall which at a certain stage becomes extremely thin and membranous. When you have 2 of these cells standing together, joining up their pits in this manner, it means that these membranes are so thin that cytoplasm can flow from one cell to the other. I'm sorry, you had a question?
Yeah, this is in all walls or just the primary wall?
We'll get to that. Just be patient. Oh, yes.
So the primary wall is inside the middle
middle lamella is simply the outer layer of the primary wall.
Okay, but the primary wall still has multiple layers.
The primary wall is what?
Has multiple layers.
Yeah, 2.
Okay.
The secondary wall will have up to 3 layers, which I'm not going into. I'm not going into S1, S2, S3. It's just a fun way to play with lignin as far as I'm concerned. Okay? So, where you have formed a pit pair, where you have formed a where you have the primary walls as thin can be, and cytoplasm can invade one cell and enter the other, these strands of invasive cytoplasm are referred to as plasmodesmata. Now, let's ask a question that was asked earlier. What if you have a system in which these cells are standing next to, these have only primary walls? What if they're standing next to a system with a secondary wall that is going to make this thick lignin undersurface? Well, while the primary wall is still functioning in this cell here to the right, it will also make a pit. And when you deposit the secondary wall, look at this, the secondary wall does not form over the pit, okay? So this means that even when a cell is dead, plasmodesmata from living cells can still enter it. This becomes extremely important when you're trying to pass water from dead xylem cells into living cortex or parenchyma cells. So even in plants, since we think of xylem as the internal skeleton of the plant, it's also the internal water pump. And the water has to go to other cells that are still alive. You can only do this by producing a pit that remains clear when you put down the secondary wall. The pit is still there, and the plasmodesmata can still invade. Okay? I realize I was being sneaky, but that's what I do. So when we look at plant tissues, we see that there are 2 types of tissues in plants. And Caleb, this is where the whole thing about is it a tissue or a colony which is the reason why it gets to be so odd, is that there are 2 types of tissues in plants. We can talk about simple tissues that consist exclusively of one kind of cell. Ground meristem, as I said, is virtually all parenchyma. And then there are complex tissues that consist of 2 or more different kinds of cells, but they need each other to form the tissue. When we look at vascular tissue, we can find up to 2 different kinds of cells in xylem. We normally find 2 kinds of cells in phloem, and epidermis, just the skin of a plant, usually consists of 2 kinds of cells working together. At functional maturity, some cells are dead, they lack cytoplasm, they consist only of their primary and secondary cell walls. So it's still a tissue, even though when it's dead at functional maturity. Let's look briefly at the first one. We have a little time to look at parenchyma. Parenchyma, from a Greek word meaning to sit beside or to pour in, these cells usually rest beside more specialized complex tissue. It's the major component of plant bodies. It's a form of padding and storage and insulation. It can be photosynthetic. Virtually all of the green you see in a leaf is made up of cells, parenchyma cells, that contain chloroplasts. Parenchyma can also be secretory. It can also perform repairs or regenerate over short distances. And because of plasmodesmata, it can move water short distances, and you can find it in virtually any part of the plant. What are characteristics of parenchyma? How do you know it if you see it? The cells are many-sided. They are polyhedral. Since there's only a primary wall, they can be compressed and mushed, and so they will have different angles. They are alive at maturity, and they are nucleated. The cell wall is only primary. It's only a primary wall. It may contain some lignin or cutin or suberin, but there's no secondary wall under it. So we should expect that these will have large pits. They're pushing their plasmodesmata all over the place, sharing it with each other if they hang in groups, or sharing it with other tissues they're sitting next to. We'll stop here today and resume this on Thursday. As for the first exam, I tend to think about this in terms of the gladiators and the earlier version, the 50s version of Spartacus, when I say good fortune to some of you. There's nothing like a classic movie. There are two